Thanks very much, and thanks to SGM and the panel for picking me. I was trying to remember the last time I won something, and I think it was Sports Day at school in 1995, so every 20 years, I think. I should also thank Maggie Smith, who's my fantastic colleague at York, who nominated me for this. And all the work I'm going to talk to you about is collaborative and involves a lot of people, but most importantly, some great collaborators and PhD students and talented postdocs who've done a lot of the work, and I'll highlight the ones that whose work I'm talking about at the end. Okay, so as an evolutionary biologist, I'm obviously interested in evolution, and microbes are really interesting in that respect because they evolve really rapidly. And the talk today is in 3 parts. So I'm going to start with how I started my career, which is experimental evolution, so watching evolution in real time in the lab. And then I'm going to explain to you a kind of collaboration I've had with some clinicians and great microbiologists at Liverpool, looking at evolution in clinical samples. And then I'm going to explain why it's important that we use those data to generate hypotheses that send us back into the lab to do evolutionary experiments where we can test hypotheses and determine causality. Okay, so I don't need to explain to this audience that microbes are important. I do to some audiences, but not this audience. And they underpin ecosystems, they underpin diseases, infectious diseases, but increasingly, as we saw in the microbiome session, they underpin our health as well. So we need to understand them, and these complex communities present a bit of a challenge, really, actually, for understanding the evolutionary processes and the ecological processes. And the really interesting thing for me about microbes is that ecological and evolutionary timescales converge, and that's because they have such big populations, microbes, and such short generation times that potentiates very high evolutionary rates. So we need to think about evolution actually as an ecological process, a really key process in microbial ecology. And probably evolution is more important in microbes than in any other taxa, simply because they can evolve so fast. And they really evolve on timescales we care about, so days and weeks. And my lab's really focused on this question. So what are the causes and what are the effects of this rapid microbial evolution? So this first third of the talk is going to be about experimental evolution, which I'm sure lots of you know about, but just to kind of recap the approach. It's essentially watching evolution in real time in the lab. And this means that we can perform really causal tests of evolutionary theory, so we can directly test hypotheses. The experiments are great because they're very controlled, so we can control the genotypes we put in, and we can determine that all of the evolution we see is due to mutation and selection, if that's what we want to determine. And we can control the selection pressures, so we can tease apart selective forces, we can disentangle complex environments. And probably most importantly of all, we can freeze the ancestors of our evolved lines, and then we can compare directly the evolved ones to the ancestral ones to see how they've changed, to see how fitness has changed, and any other trait that you might be particularly interested in. So just to really hammer this home, the key thing about experiments and doing experimental evolution is that we can determine causality, and we can disentangle complex environments and pick out the really important drivers of natural selection in those environments. So my lab is interested in interactions, species interactions. And as I showed you with those complex communities, there are loads of species interactions going on in microbial communities. And this is just a kind of cartoon of the subset that we might be interested in. So bacterial species, or bacteria and fungi even, compete with each other. Phages predate bacteria, protists also predate bacteria, and then bacteria can share genes through mobile elements, and those mobile elements themselves sometimes have their own evolutionary agency. So in today's talk, I'm going to focus on bacteriophage interactions, but just to plug members of my group who are also here today over the next 3 days, we work on other things too. So if you want to learn more about our bacterioplasmid work, then 2 postdocs, Jamie Hall and Ellie Harrison, are giving talks, and Charlotte Stevenson's giving a poster. And if you want to learn more about our bacteria-protist interaction work, then Megan Sorensen's also giving a poster. And I think Megan and Charlotte are very brave master's students for turning up to a big conference like this and giving posters. So phages are important. They're everywhere. They probably outnumber bacteria by an order of magnitude. So that means we've got, if you believe Roger Hendricks, 10 to the 31. Really loads of them. They're everywhere. And that means that they're a really pervasive selective force. So they kill bacteria. They're a major cause of mortality in most natural environments. What that means is that they have important ecological effects. So people think that phages are really important for maintaining biodiversity in microbial communities. But they also have really important evolutionary effects. And they're a really strong selective force in natural environments. So we really need to understand the interaction between bacteria and their phages. And as an evolutionary biologist, these kind of interactions, these antagonistic interactions between species are really interesting. And the reason for that is laid out in something called the Red Queen hypothesis, which was proposed by an evolutionary biologist called Lee Van Valen in the 70s. And the Red Queen hypothesis really puts biotic conflict, so these antagonistic species interactions, at the very heart of evolution. And so, because these antagonistic interactions select for repeated cycles of adaptation and counter-adaptation, so in this case, infective phages impose selection for resistance, which then imposes selection for new infectivity. So we get these endless cycles of evolutionary change, counter-adaptations met by counter-adaptations in the interacting species. And that gives us a requirement for continual evolutionary change just to remain as fit as you are today. So you have to these species have to keep changing in order just to maintain their fitness, not to get any better, because the biotic environment's constantly degrading. And so if you're interested in that, we wrote a review for Prop B recently, which kind of sums up that field. And what we were interested in is quantifying the Red Queen effect. So how much evolution do those coevolutionary species interactions actually drive? And so one prediction of the theory would be that you'd expect elevated evolutionary rates at genes involved in these antagonistic interactions. And we tested that with a really simplistic experiment. So we allowed one set of populations to just co-evolve. So we did batch culture and we grew bacteria and viruses and then transferred them to fresh media and then again and again, et cetera, et cetera. And then as a control, we decoupled the evolution of the virus and the bacteria and we just allowed the virus to evolve. And we did that by separating the bacteria and virus at every transfer and replacing the bacteria with the ancestor grown up from a frozen stock. So here, just the virus is evolving, and here both the bacteria and the virus are evolving. But the population sizes are exactly the same, the number of generations is approximately the same. What we did at the end of the experiment was then sequence the entire population of virus from every replicate of that study. And we did this with Pseudomonas fluorescens, which is just a plant-associated soil bug, and a lytic phage with a 40 kb double-stranded DNA genome. So just to begin with, what we saw phenotypically was that those two regimes selected for different traits. And so here we're plotting infectivity range against some measure of fitness, the number of doublings, how fast they grow. And we can see that the coevolution treatments selected for increased infectivity range, but no change in growth rate, whereas the evolution treatments selected for an increase in growth rate and no change in infectivity range. Okay, so we know that phenotypically these selection regimes have caused different things, and in the direction we'd expect. Things that co-evolve increase their infectivity range, things that just evolve are increasing their growth rate. So when we look at the genomes of these viruses that have evolved and plot a genetic distance tree, what we see is really interesting, I think. So this is the ancestral virus, and then we can see that the two treatments cluster together. So these are the evolving viruses, these are the co-evolving viruses. And so I want you to take away from this is that the branch lengths over this side are about twice the length as they are over here. So coevolution is accelerating the rate of molecular evolution. It's doubling the rate of molecular evolution in this study. We also see that they're kind of clustering together, the two treatments. So the evolved ones are evolving in the same direction, and the co-evolved ones similarly are evolving in a different direction. But also, the distances between the independent populations are much larger under coevolution. So coevolution not only selects for more evolution, it selects for more divergence between populations. So next we were able to link that genetic evolution to phenotypic evolution. And so all we're doing here is taking bacteria that we've isolated from these populations and exposing them to the viruses from the end of the experiment. So here we've got the viruses here. And if it's red, then the virus can infect. If it's grey, then the virus can't infect. And so we see that they're different. And if we draw a tree of those phenotypes, then we can see that that has the same structure as the genetic tree of the co-evolved genomes. So 1 and 3 cluster together here, and they cluster together here. And similarly, 5 and 6 are out here on the genetic tree, and they're out here on the phenotypic tree. So that suggests we've got quite a tight link between genotype and phenotype in this virus. And we can go and delve into the genome and figure out where those mutations are happening. And if we do that, and we plot it, then what we're plotting here is the pairwise distance per gene, and we're plotting that against the genomes. This is the phage genome along here. What we're showing here is the intensity of those mutations within, or rather the frequency of those mutations within each population. And then we're plotting the averages here for each gene. And so in pink, we've got the co-evolving populations, and in blue, we've got the evolving populations. And so what we can see is that for 4 genes, they actually evolve faster under coevolution than they do under evolution, suggesting that they're involved in this interface between the bacteria and the virus, and they're involved in evolving broader infectivity ranges. So those 4 genes are the tail fiber, which is this structure here, which is involved in that initial step of binding between the bacteria and the virus. That's this one here, which is evolving loads more in the coevolution treatment. Another one is this structural protein, which is here. So we think that this is in this base plate region, so it's also involved in attachment. And here we get a lysine insertion, which causes a massive expansion in host range. And then 2 internal virion proteins, which we don't really know what they're doing particularly, but they are evolving more under coevolution. And then we get 1 gene that's to do with adapting to the lab and evolves in both treatments. So using quite simple experiments and a bit of genome sequencing, we can show that there is a Red Queen effect. So coevolution accelerates molecular evolution. It doubles the rate of substitutions. Coevolution also drives greater between-population divergence. So it's important in terms of generating diversity in the world around us. And there's also this tight link between genotype and phenotype, and we can tie infectivity evolution down to 4 genes in the virus. And so anyone that works on real systems might be sat there thinking, okay, fine. What do we really care about the evolution of viruses in test tubes when all we really want to know is what's going on in these important environments? And so who cares? You know, who cares about these unnatural test tube environments? And I have some sympathy with that. But what I think we can do is actually start to apply the techniques that we develop in terms of the experimental design and the analysis in experimental evolution and apply them to these more important systems in the real world where we really care about what's going on. So where we've tried to do that is in collaboration with Craig Winstanley and Steve Patterson and some very nice clinicians at the Liverpool Chest and Heart Hospital, is to look at the cystic fibrosis lung. So CF's a hereditary disease. It's relatively common amongst Caucasians, so maybe 10,000 people in the UK are affected by this. So it's a mutation that causes sticky mucus in the lung and other body sites, and these people are very prone to lifelong infections of Pseudomonas aeruginosa. These cause morbidity and they cause mortality amongst those patients. And there's lots of anecdotal evidence that there's lots of diversity among these populations, and there's now increasingly some genetic evidence too, and I'll show you what ours is in a minute. And the problem here is that diagnostic labs don't actually take that diversity into account very often. Some diagnostic labs do, but most don't. And what they do is pick a single colony or maybe 2 colonies that look different and then they use those for their antimicrobial susceptibility testing. So actually understanding this diversity that evolves in the lungs could have really important clinical consequences and suggest some very simple things that NHS labs could do to improve their diagnostics. So what we're asking in this project is how diverse are the populations, how dynamic is that diversity in time, and also, does it matter? I mean, are these actually diverse in traits that we care about? So we've been working on something called the Liverpool epidemic strain, which first cropped up in the mid-90s when there was a sweep through this children's CF unit in Liverpool of a ceftazidime-resistant strain. It can superinfect, so that means it can infect an already infected patient and displace their Pseudomonas aeruginosa genotype. It spreads to parents, which is unheard of really in cystic fibrosis. It swept to very high prevalence pretty quickly and then spread to the UK. And then in 2010, it was the first reported incidence of this strain in the US. And it was genome sequenced by the Winstanley group in 2009. So what's important here is that this caused an epidemic. And so what that means is that we have a natural experiment, and it's almost exactly the same as the kind of evolutionary experiment I might design in the lab. What we've got is a load of patients who are all infected with approximately the same strain at roughly the same time during an epidemic. And what we've been able to do in collaboration with the Liverpool Chest and Heart Hospital is to sample these patients every time they come to clinic since 2009, which means that their infections have had some time to diverge since their original infection, which was sometime between the late 90s and the early 2000s. And now we're tracking the dynamics of the diversity within their lungs. So just to give you a feel for the sampling we do, we've got 10 patients, and this is just the first year of sampling. And we get a sample whenever they come into clinic, so when they're stable, which are these yellow samples here. And we also get samples when they come in with what's called an exacerbation, which is when their symptoms are very severe. And we get a sample when they're first admitted to hospital, when they're being treated with IV intravenous antibiotics, and also when they've recovered from the exacerbation. And what we do is pretty simple, really. So we take the sputum samples, we plate them out on Pseudomonas selective agar, and we pick 40 colonies at random. So this is ecological sampling. We're trying to sample the diversity in an unbiased way. And then we expose those to a bunch of phenotypic tests. We test them for auxotrophy, hypermutability, toxin production, a quorum sensing produced LASE, quorum sensing regulated, LASE, and antibiotic resistances. So this is a protease. And what we also do is sequence the genomes of those 40 colonies. So just to start with the phenotypic diversity, then what we're plotting here is diversity against time, and we've got each of our patients. And what we can see is that diversity is always high. So this is just looking at that set of phenotypes that we've picked, and we're putting those into haplotypes and saying, how many haplotypes are in each sputum sample? And with the exception of one sample here, it's always really high. So you can pick 40 colonies, pick 2 at random, they'll be different phenotypically. So that phenotypic diversity is important, and the reason is things like this. So this is a load of antibiotics that they use in the clinic. This is data from a slightly larger cohort, but including some of the same patients. Data from Joe, Fothergill, and Craig at Liverpool. And what we're plotting here is the zone of inhibition against patient. And then the red line gives you when you'd consider it to be clinical resistant. So anything above the line is sensitive, anything below the line is resistant. And you can see that within individual sputum samples, there are very resistant and very susceptible things coexisting. So that phenotypic diversity is actually really important, because you might think, if you picked one colony, that you're going to choose the right antibiotic, but actually half the population are completely resistant. And that doesn't just hold for one antibiotic, that's holding for 4 of these really important antibiotics that they use in the clinic. So when we look at the genomes, then we do see lots of genetic diversity within single sputum samples. So most of the diversity, when we look across the whole set, is within sputum samples. So this is just a single sputum sample from patient 3, where we've genome sequenced every single of the 40 isolates that we took. And this really surprised us, this data. What we've got are 2 coexisting lineages that are really quite highly diverged. And those lineages themselves are quite diverse. So there are a lot of genotypes within each lineage. But there's a huge amount of genetic distance between these 2 coexisting lineages within this patient. By this sort of approach, you can also pick up things like recombination. So this is a very powerful way of figuring out what diversity is in your lung, and it shows that it's actually really important to sample more than one colony, and the more you sample, the more diversity you're likely to find. But next we wanted to know if this pattern of highly diverged but coexisting lineages within patients is common. So this pattern suggests that these haven't diversified within the lung. It suggests maybe that there's ongoing transmission going on. So that's what we needed to figure out. And so to do that, we had to expand across more of the patients. So this shows you a phylogenetic tree of all of the lineages that we detected across all of the patients where we've done genome sequencing of their isolates. So that's nine patients. And so what's going on here is there are a bunch of lineages within this population of the Liverpool epidemic strain, which have all diverged from the most recent common ancestor, which was presumably the strain that caused the epidemic. And each lineage is separated by some SNPs. So we call these lineage-specific SNPs along these branches. And then there's some diversity within that lineage. So that lineage has then diversified within patients. And what we see is that diversity is the rule, and more than that, coexistence of diverged lineages is the rule in the LES. So 7 out of 9 patients are co-infected by divergent lineages, and only CF6 and CF10 contain single lineage infections which have presumably infected once and then diversified within the patient. And we see this evidence of transmission. So by zooming out across a cohort that all attend the same clinic and doing the approach that we've taken, we can see 2 patterns which suggest that there's ongoing transmission. So firstly, those coexisting, co-infecting lineages within patients are quite often much more different from each other than they are to lineages in other patients, suggesting that they've been acquired horizontally by transmission. So to give you 2 examples, here's CF3 and CF3, so really highly diverged. And here's patient 5, again, contains 2 lineages that are very highly diverged, suggesting that they've been independently acquired. And then amazingly, considering we're only sampling 9 patients here among a clinic of hundreds of patients that attend the Liverpool clinic, we actually see lineage sharing among patients. So here's some examples. CF7 and CF9 share lineages. There's another 9, CF9, CF7 there. And similarly, CF1 and CF4 share lineages. So presumably these patients are exchanging lineages, which was amazing to detect. And this suggests that even though the clinicians at Liverpool put a lot of effort into stopping transmission between patients, they're quarantined, they never see each other in the clinic, actually transmission is going on anyway, and that's probably because patients meet each other socially. So our next question was, well, what genes are evolving? And the most powerful way of detecting genes that are under selection in these sorts of studies is to look for parallel evolution. So that's genes or operons or functions that are being targeted independently by selection across the multiple independent lineages. And that's a very good signature that selection is operating at those loci and they're involved in adaptive evolution. So what I'm showing you here the concentric rings are each of the lineages that we've identified. And then the blobs are mutations that we've detected that have evolved in parallel among lineages. So they've been independently targeted by selection across multiple lineages. And taking this approach of filtering out all of the unique mutations and just looking at the parallel ones gives you the footprint of selection. So it tells you what selection is operating upon this population of the Liverpool epidemic strain. So what we see is a lot of antibiotic resistance, and that you might expect. They get exposed to a lot of antibiotics in the treatment of these infections. But the diversity of antibiotic resistance mutations is quite high. So all of these ones in red are antibiotic resistance mutations that are evolving in parallel across lots of lineages. Secondly, we see evidence of the transition to living in gloop, effectively, the transition to living in sputum, which is basically saying that they need to convert themselves into a biofilm-dwelling organism. And we can see mutations that affect alginate and biofilm production, similarly here, an alginate producer, and here, again, alginate. And then the third kind of adaptation I want to highlight as secreted products, or rather the regulation of secreted products as well. So pyaverdin, which is an iron-scavenging siderophore, seems to be lost in 3 of our lineages, and LASR, which is the global regulator of quorum sensing, is also lost across multiple lineages independently, suggesting selection against quorum sensing in the lung. Okay, so next, I want to talk a bit about dynamics. So it's all very well to say that these populations are very diverse within patients. But so can you basically characterize a patient for their lineages and then say, okay, they're characterized, we don't have to worry about it, or does this change through time, and is it highly dynamic? Well, what we see is that it's highly dynamic. And so there's 2 lines of evidence for this. Firstly, phenotypic evidence. So again, going back to our phenotypic data, and we're plotting the sharing of haplotypes. And we're plotting that, and this is for pairwise comparisons, and then we're plotting that against the time between those 2 pairs of samples. And so you can see that for most patients, there's a negative relationship there. So the further apart in time 2 samples, sputum samples, were collected, the lower the probability that they share haplotypes at these phenotypic traits. And we can address that question with genomics too. And so for all of our patients that have multiple lineages, we've looked at the structure of those populations in terms of the lineages they contain and the abundance of those lineages in multiple samples. So again, we've got time along the bottom, and we've got patient identity here. And so what you can see is that there are some incredibly dramatic changes in the abundance of lineages over time, but also the identity of lineages over time carried by patients. So one interesting example is here. So this is a patient that suffers an exacerbation. This is their acute phase where they're coming in very sick. This is antibiotic treatment, and this is recovery. But we can see that in the closely sampled in time, stable sample before, they were mostly the red lineage, and then when they were feeling very sick, they were mostly the green lineage. And similarly here, there's a change in the lineage structure as they go from stable to exacerbated, suggesting that these shifts in population structure are actually quite important for driving the clinical symptoms that we see. Most remarkably, we see exchange of lineages between patients. And we did not believe this data to begin with. So CF09 and CF07, we can see that they basically exchange lineages. And so we've interrogated this now with qPCR and other approaches to figure out what's going on. And it does look like it's real. These patients are switching lineages. So I think that's pretty amazing. And it's not just those patients either. So CF04 has a complete turnover of lineages in really a short space of time. So in the matter of a month and a half, 6 weeks, they go from being predominated by this red and magenta lineage to being predominated by these green and blue lineages. So I think this is amazing. So we've got enormous genetic change occurring over relatively short periods of time in these patients, and presumably having really important clinical effects. And we have some evidence for that, which is that if we go back to the phenotypic data, then we can see an association between piocyanin production and the exacerbation. So patients suffering exacerbation have an overrepresentation of high piocyanin producers in their lung. And this is a toxin, so it sort of makes sense that that might be driving the exacerbation. But obviously, with these kind of data, you can never assert causation, really. It's all correlational. Okay, so just to summarize that second part. So these single-strain bacterial populations in CF are really highly diverse, and that diversity is highly dynamic in time. Most diversity is present in the single sputum sample, so if you partition the diversity across lots of patients, you'll see the bulk of it is within patients. The diversity occurs in clinically important traits like antibiotic resistance, like secreted products, like toxins, like quorum sensing regulators, and also those genes that control biofilm formation. And so using genetic data, you get this much higher resolution picture of what's going on. And it's allowed us to detect parallel evolution. So we now kind of can see the footprint of selection in the CF lung on the genomes. And also, it's allowed us to detect patient-to-patient transmission in the clinic, which is actually a really important clinical finding that we can feed back to the clinicians and help them care for their patients even better than they already do. So for the last part of the talk, this is part showing you data and part call to arms, really. So I think what we need to do is take data like that and other lovely data that I've seen earlier this morning, where people are sampling enormous sets of strains, and use those to generate hypotheses about selection in these environments and go beyond correlation. So all we can ever do in these environments is detect correlations between selective effects or hypothetical selective effects and hypothetical selective causes and the effects that they have. And we can never really get at causation. To do that, we really need experiments, and we need to design really better experiments to try and get at that. So I'm going to give you an example of a controlled selection experiment that we've done inspired by our work in the CF lung with the Liverpool epidemic strain. So this is a question that's bothered me for ages, and I know it's bothered other people in this room, like Freya Harrison and Steve Diggle and people like that. What's actually driving evolution in these lungs? So what are the selective drivers that operate in the CF lung? And how can we disentangle them? So we might think that we know everything. We might think that it's just antibiotics, obviously. But it might be the interaction of antibiotics and other factors in the lung that drive the antibiotic resistance, or at least potentiate it. So it might be immune responses, the application of drugs, species interactions in the lung could be a really important driver of evolution in the lung. And I showed you before that species interactions actually accelerate evolution, so they might actually be very important here. And you can go on and on and on, dreaming up other sources of selection that might operate in lungs. And it's only through experiments, I think, that we can start to disentangle those and really pin selective causes against their mutational effects. And the way we do that is by experimental evolution in this stuff called artificial sputum medium. So this is gloopy stuff that's physiochemically similar to sputum. And the most important thing is that the bacteria grow in these floating microcolonies. And this actually looks quite a lot like CF sputum. And Craig and Joe Fothergill have gone on to actually do some animal model work as well, which I won't talk about because it had nothing to do with that. Okay, so we use this ASM medium as a proxy for lungs. And the question we're interested in is what effect phages have on evolution in the ASM. So one of the interesting things about the LES, so the Liverpool epidemic strain, is it contains 6 prophages in its genome. And all of those prophages are active, so they're not remnants. They're not just bits of prophages that have fallen apart. But when you look back at the genome sequence of the LES, the first isolated strain, those phages can be induced out of the genome to lyse the cell, so they're all active. And actually, if you do some site-directed mutagenesis of phage functions and chuck those strains in a rat model, then they're actually really important to the competitiveness and fitness of that bug in the lung. So the phages could be playing a really important role ecologically, and also could be playing an important role evolutionarily. And some evidence for that comes from this data, which is showing you the dynamics of the bacteria, so Pseudomonas aeruginosa LES in the lung, and the phages that infect it. So we've just got log population density here, and time in days, and the solid line is aeruginosa density, and the dotted line are free viral particles. So these are viruses that have lysed the host and are sitting in the sputum as virions. And so you can see that viral densities are 10 times, 100 times higher than bacterial densities in these infected patients. And it holds across all of these patients. And this data actually hides a lot of complications because there's actually multiple phages in there. There's 6 of them, as I said, which we've added up to give us this one line. The other interesting thing is that the densities of bacteria and phages are variable through time. But the analyses we've done suggest that these viruses are being produced just through induction of lysis in bacterial hosts that have the prophage, but also that lysis might actually play some role in controlling population densities of the LES. Okay, so what we did is just design a simple experiment again. So we're evolving populations in the artificial sputum, and in one treatment we've just got Pseudomonas on its own, and then in the other treatment we've added 3 of the LES temperate phages. And it's really simple, this experiment. We just put the bacteria in and then chucked in viral particles. That's all we did. And then we passaged that for 250 generations and looked at what happened. So firstly, did the viruses lysogenize the hosts? Yes, they did. So this is just showing you the 6 replicate populations, and then various forms of lysogen. And all I really want you to take away from this is that lysogen's fixed in all but one of our replicate populations, and they're almost always contained 5,4. And 5,4 is really interesting, as I'll show you in a minute, because it's a generalized transducing phage, and it can insert seemingly just about anywhere in the genome. Okay, and we see some establishment of polylysogeny here as well, which I think is nice and quite interesting, and it's variable between populations. So did they evolve, is the next question, and yes, they did. So here we're looking at a thing called the selection rate constant, which is essentially if you directly compete the evolved strain against its ancestor and then measure selection. So how much fitter is the evolved strain relative to the ancestor? And we're doing that for our control treatment with no phages and our phage treatment. So here it's against the ancestor. The ancestor is phage sensitive, and we can see that both treatments have high fitness, very high fitness. By contrast, if we compete them against an isogenic pill A knockout, which can't make the pilus, and all of these phages infect via the pilus, then we can see that they're still fitter. The evolved ones are still fitter, but it's a much smaller effect. And this speaks to the bacteria using the phages as a weapon. So they've got these prophages, they can release the prophages and kill competitors. And if you're interested in that story, then my postdoc, Ellie Harrison, had an amazing paper in Evolutionary Applications that you should read about polylysogeny. Okay, so there's some ecological effect of having prophages, but is there an evolutionary effect, is the next question. So we did genome sequencing, and we used the same protocol that we used for the CF sputa. So we pick 40 colonies, we sequence them, so we get some sense of population diversity and the complexity of those populations. So here's the data from this experiment. So instead of lineages, this time we've got the replicates. So in effect, they are independently evolving lineages. The concentric circles are the replicates. And we've got the control treatments with no phages and the phage treatment treated with phages here. And then color-coded, we've got the effects of the mutations. Basically, we've highlighted things that have affected virulence-associated traits, but there are also some metabolic things going on that I've not highlighted. And then we've just coded up different kinds of mutations, and the ones I want you to focus on, or at least notice, are these ones caused by prophage insertions, which are these little sticks with bars on the end, like a little dumbbell. Okay, so what we see in the absence of phages is adaptation to the ASM. And the way that they're evolving is by changing their biofilm characteristics, which are these blue mutations, but also losing the flagellum. So we see quite strong selection against the flagellum in the ASM. On these plots, the size of the blob is telling you how many of the 40 clones that we sequence have that mutation. So it's telling you something about how close to fixation those mutations are in those populations. And again, all I'm plotting here are the parallel mutations. So what we're trying to detect are these loci that are being independently targeted by selection across independent lineages that are evolving separately. So lots more mutations than just these occurred, but these are the parallel evolving ones. So in the absence of phages, we have a pattern of selection which looks like it's about evolving to live in gloop. And it suggests that losing the flagellum isn't anything to do with immune exposure in Pseudomonas aeruginosa growing in gloop. It's to do with just growing in gloop. So more exciting than that, we've got the phage treatment over here. And what you can see is that it's selecting for different types of mutations. So adding phages changes the trajectory of evolution. So the different characteristics or traits that are being selected in the presence of phages is loss of the pilus. So this makes sense because the pilus is the site of infection and binding of these phages. So what is being selected for is basically resistance to phages. The really cool thing is that it's being caused by prophage 4 insertions. So the phage is jumping in to the target and disrupting pilus, the making of the pilus in this strain. And essentially it's slamming the door behind it and stopping the other phages getting in. That's happening in some of our replicates, we think for sure. The second really important and interesting trait that I want to highlight for you is quorum sensing over here. So we don't see any quorum sensing mutations evolving in parallel in the controls, but we see really parallel evolution and parallel loss of quorum sensing in the phage-treated populations. So we're seeing loss of LASR, both by mutational SNPs, but also by phage insertions. And we're seeing loss of MVFR, mainly via prophage insertions. So this is pretty interesting, I think. So there's a big literature about loss of quorum sensing in the CF lung. And this data suggests that phages might be implicated, or temperate phages in particular, might be implicated in driving the loss of quorum sensing. The reason that might work is there's a fabulous paper by Gosch and co-authors in 2009 which showed that prophages can be induced by AHLs, so the molecules that bacteria make which allow them to quorum sense. And it suggests that phages can somehow sense the population density and then lyse the cells when their host is at high population density. So you can see, if you're in a community with loads of phages, and most of your bacteria have got prophages in their genomes, that it might be a good idea to lose quorum sensing, because that's going to basically prevent your phages lysing you. So they're not going to be able to sense, oh, we're at really high population density, I should kill my host. If you're not quorum sensing, then you don't get killed by the phage. And we don't have that pinned down mechanistically yet. But the really nice thing about the Gosch paper is that they looked at soil extracts and water extracts and just applied AHLs and saw loads of phages being produced. So it seems like it's a pretty general effect. Okay, so phages drive the loss of quorum sensing, which I think is a nice punchline. So evolutionary adaptation to ASM is being driven by the loss of motility, especially the flagellum. It's also being driven by metabolic and biofilm changes. Temperate phages, adding temperate phages, changes the trajectory of evolution, and it also changes the mode of evolution by causing mutations. So phages select for, in parallel across multiple replicates, loss of type 4 pilus, which is the binding site of the phages, loss of quorum sensing, and as I explained, AHLs induce phage lysis, and loss of type 6 secretion systems, which I haven't really talked about. Okay, and then this important effect that phages cause mutations. So this phage, prophage, this phage 4, is inserting in lots of different sites across the genome and causing beneficial mutations. So just to skip back, it's inserting in pilus-associated genes, it's inserting in these not well annotated genes, it's inserting in these quorum sensing genes and these quorum sensing genes. So they're causing beneficial mutations across the genome. Okay, so that brings me to my final slide. So I hope I've convinced you that rapid evolution is worth caring about and that it's a fundamental process of microbial ecology, and we really need to start factoring it into our understanding of microbial ecology. These rapid evolutionary dynamics have important clinical consequences. I'm sure they also have important environmental consequences in any environment you wish to care about. But here I've talked mainly about clinical consequences. The key thing is that evolution experiments allow causal tests of hypotheses. And what we need to start doing is linking up genome studies of clinical samples with experimental tests so that we can go beyond causation, beyond correlation, and start testing causation and trying to understand the selective environment of infections. And to do that, what we need is collaboration. So we need people like me, evolutionary biologists, to talk to people like you, microbiologists, and with clinicians too, to really kind of get full understanding of these systems. Which just leaves me to thank a lot of people. I only had space really to list the people that were involved in the work that I've talked about. The cystic fibrosis group is big and ever growing. The key people I'd like to thank are Dave Williams, who did lots of the genomic analysis. He's a fantastic bioinformatician, if anyone's looking for one. Emily Davies, a PhD student, and Chloe James, a postdoc, now lecturer at Salford, who did the ASM experiments and also the phage in lungs experiments. And Craig Winstanley, who's been my long-term collaborator there, and Steve Patterson, similarly, in Liverpool, who've been just great to work with, fantastic people. And the two clinicians, Martin and Martin. And then the bacteriophage coevolution work, the experiments were done by Tom Vogwill, who's a PhD student and now is at Oxford as a postdoc. Steve, who's a sequencing whiz. Angus, who's been a long-term mentor and collaborator of mine. And then finally, I need to thank a lot of people who've helped me have the career I've had. So supervisors, my PhD supervisor, Paul Rainey, postdoc supervisor, Michael Hochberg, mentors, lots of people still from the Rainey lab who I'm still working with now, like Rob Jackson, who's here, and Angus Buckling. Colleagues at Liverpool and also at York. I used to work at Liverpool. Collaborators, lots of them, but most importantly, all the students and postdocs who've done all of the lab work, because I haven't been in the lab for about 10 years. Okay, so everyone past and present, basically, but thank you very much for listening.